Alison has already mentioned Rich, who did a PhD and was working with Jim 1990 to 94, I believe. He will tell you about some of the work that he's been doing in industry, and you've been in a couple of companies. Are you after? I think maybe that. Just check it works. A bit closer. That's Alison's. Let's go for that one. Or that one. Or that one. Hey, where's Finian?
Just press buttons.
I've pressed every button I can.
Have you plugged in the HDMI?
No, it's a USB-C.
It's a USB-C, but it isn't. Maybe it's thinking about it.
There we go. No. It's useful. Do we have HDMI? Oh, right. Let's try that instead then. Mount that. Hey, we're in. Excellent.
Let's get both screens going.
Awesome.
Brilliant.
Right.
Over to you, Rick.
Great. Thank you, Helen. Wow. So, yeah, well, thank you for the introduction. And yes, I was a PhD student in Jim's group from 1990 to 94. I was also a student at St John's, an undergraduate student at St John's, where Jim was my organic chemistry supervisor, much like Finian, for the final 2 years of my studies. And Jim also interviewed me for my place at St John's when I was 17 years old. So I've had a lot of that early period of my academic career. I had a lot of interactions with Jim. And actually, probably in education, I spent more time with Jim than I had with anybody else in teaching over the course of my life. So I'm obviously amazingly grateful to him for investing his time in me and giving me the opportunity to go out and do things that I've really enjoyed and sort of pursue things which really interest me across the course of my career. So what I'm going to talk to you about today is some recent work we've been doing at GSK about making oligonucleotides. Now, I've been at GSK for nearly 8 years now, but I think this slide is actually something which is consistent across our industry as a whole, across the pharmaceutical industry as a whole, and it's something that I found when I was working at Dr. Reddy's beforehand and Chirotech, that the drug molecules we're being asked to make now are increasingly complex, but we're also under immense pressure to make these complicated molecules more efficiently, more cheaply, so we can get greater access to larger, increasing patient population, and also we can make our molecules more sustainably. So one of the things that we have been doing is employing what we've got on this slide here as being new technologies. None of these are particularly new, using catalysis and the likes, not really, but I think recent advances have made things like biocatalysis, where I have been working for pretty much all of my career since I moved back to the UK in the late 90s, viable options. In terms of biocatalysis, when I first started working with enzymes, we had very limited application or classes that we could use, but then now we've moved to the point where with the likes of directed evolution, then we're able to tailor our catalysts to a very specific reaction at any point. And actually, unlike using transition metal catalysis, I think we can optimize very, very specifically for a given application. So this is incredibly powerful, and we are now at a point where there are certain reactions carried out in industry where the biocatalytic option is the preferential one. So applying this to oligos, there's a lot of amazing structural complexity in all nucleic acids, and really using this slide, which is to show the differences you can get in DNA and RNA, but we have a number of different areas where you can introduce diversity, even in naturally occurring molecules. You have your sugar, you have your nucleobase, and you have the backbone. Now, when we move into therapeutic oligonucleotides, then we can start having chemical modification here, we can have chemical modification here, and we can have different backbones as well. So instead of just phosphates, we can have phosphorothioates or methylphosphonates linking our ribose units together. We can have odd chemical substituents on the ribose units, and we can also have functionalization of our bases as well. So even though these are linear polymers and they are very modular, it's very easy to quickly build in an awful lot of complexity into these molecules. Now, because of the linear modular assembly, actually it lends itself very well to some chemical approaches. And the solid phase approaches of making them has been, a standard for 30 odd years now. And it's quite a nice looking cycle, really. You start with your unprotected nucleotide, which is linked to a solid support. You undergo a coupling reaction to extend your chain by 1. Any synthesis failures you can cap off here, and then you oxidize this phosphorus 3 up to phosphorus 5, deprotect, and you return to the new cycle. If you've made your compound, then you cleave it off the resin and you end up with an oligo which you can purify. So this has been highly optimized, and it works actually really well. If you're making a compound which has got 20 nucleotides in this, it's actually about 80 steps of chemistry on the solid phase, and you've got your cleavage and deprotection, so maybe we'd say 82 steps of chemistry, which is an awful lot for an API. I think the most complicated small molecule APIs around at the moment are made through about 60 steps of chemistry, and they're not linear sequences, they'll be convergent sequences where the longest linear sequence is probably about half that. If you take this cycle for a 20mer with a chromatographic purification step at the end, the yield that you can get is often around 50%. With the small molecule syntheses, which are shorter, those yields will be below 10% end to end. So this is actually a really, really efficient way of making oligonucleotides. But there are issues with it. We have large protecting groups, both the tritile groups and the bases are often protected during the synthesis. So we do have issues with atom efficiency. We have this oxidation step here, which uses iodine, adds complications to waste. And overall, this is an incredibly solvent-intensive process. So we use a lot of acetonitrile and a lot of toluene in doing this kind of chemistry. So what do we want to move towards? We'd like to move towards something where we could use standard plant equipment to do this. So we could do some more chemistry in stirred tank reactors. Another reason for doing that is actually that oligos are now no longer for niche application. There are some ton-scale oligos that have been approved. And with the solid phase approach, you are somewhat restricted by batch size. So on the solid support, you probably get a batch size of 6 or 7 kilos, maybe 10. So if you need to make a ton a year, that's a lot of batches you've got to do. So we'd like to be able to work with larger batch size. But coming back to greener and more sustainable chemistry, we'd like to use less solvent. Actually, if we do the process mass analysis of oligosynthesis for 20mers, in this paper here, they do an analysis using kind of a standard 20mer as such. I think they arrive at the conclusion that the PMI is about 4,300. That's 4,300 kilos of waste per kilo of product. About 3,000 kilos of that is probably acetonitrile. So we can really do much better in those areas. And if we are more efficient, then we have the opportunity to severely reduce the cost of these medicines. So we've been looking at using enzymes to turn these syntheses from a linear sequence into a convergent sequence. And this is the approach that we've been employing, where we start with our short fragments. And at this stage in time, we have been making these chemically, but we do see that ultimately they could be made enzymatically, and we could see an end-to-end process all in water. These strands we align on this complementary template made of DNA. So we end up with a duplex, which has got 2 nicks in it. And this is where the enzyme comes in. We use a ligase to seal these 2 nicks and generate a full-length duplex. Now that all looks great. We have to get our product, which is the orange bit, we have to remove that from the duplex, from the template. We also have to consider other things which will be in this reaction mixture. So we have to consider the intermediates that we'll have in this, and we also have to think about unreacted starting materials and some of the impurities that are carried through from the synthesis of our precursors. Now this, for a process chemist, is quite a mixture to deal with. And these are large molecules, so applying traditional methods like crystallization to get to our final product is probably not going to work. So we need to think slightly differently about how we're going to do this. And this is our approach. We've decided that we can employ filtration to actually separate everything out. To make this work, we take our templates and we array them around a central core and really boost the molecular weight of this species. This is something we now call a hub template. And in our lab experiments, we've been working with things which weigh about 50 kilodaltons. We then have all our materials duplexed with this template, and we take advantage of Watson-Crick base pairing to selectively denature things from this template to melt them off the template. And based on GC content and the number of base pairs, you find that the lower molecular weight things can release before the higher molecular weight things. So with this kind of reaction, as it happens, our desired product is always going to be the highest molecular weight species, so that gets left on the template last. And we can remove everything first, and then go to our final isolation purification step and get to our final product. In terms of a process outline, for the enzymatic chemistry, we can do this in a stirred tank reactor. We then, at the end of the reaction, we remove the protein by heating, and this denatures it, and that can then be removed by filtration to give us a nice solution which comes into ultrafiltration. And the ultrafiltration is a 3 or 4 step process, really. You have an ultrafiltration phase first, which concentrates the mixture. We do this so we really minimize the amount of water used in purification. We then change the pH from the reaction pH, we elevate it slightly, and this then starts melting off the intermediates and the shorteners, which can be removed by ultrafiltration, leaving us with our product stuck to the template. That is removed at a different pH to give us something which can be desalted and lyophilized. And then to make this really work, we have a lot of time and effort invested in making our template, and making our hub template. We recycle it. So it's left on its own in solution. At the end, we return it to the reaction pH, and it can go straight back into the vessel, be used again. How we develop these reactions is pretty standard for biocatalytic reaction development. We always start off working at a very small scale. Reactions carried out in 96-well plates, and this is how we do our protein engineering to get the catalysts that we require to do the chemistry, but it's also how we start developing the initial parts of the process, understanding the reaction conditions we want to run at, for example, the pH or the temperature of the reaction and that kind of thing. And we do this all, very small scale in sort of 200 microliter to 1 mil type chemistry. Maybe specific to these processes, we do actually have to work on a certain scale at some point to provide material to go into the downstream process development. It's not really feasible at this stage to develop downstream process on microliter scale, so we do need to work at a slightly larger scale. But that's great because it actually gives us the opportunity to isolate a product for the first time. And from that, we can start looking at the type of impurity profile that we have, and therefore we can start thinking about working with our suppliers to set the specifications for the shorteners to go into our enzymatic ligation to give us a product of desired quality. And from there, once we've got this set, then it's a question of validating the process in larger scale in the laboratory before we're finally ready to transfer this out to pilot and work on up to kilo scale. So we've had the opportunity, or I've had the opportunity to do this with an antisense oligonucleotide. And this is a 20-mer. It's what we term as a gapmer. So in the green nucleotides, these are all DNA bases, and they're flanked at each end by 2 MOE protected nucleotides. Now, when we set our disconnection strategy for this, we selected the 767 approach to allow us two things. One, to not challenge the enzymes particularly much, because this molecule is fully phosphorothioated, so we have PSs all across the backbone, and we didn't want to challenge the enzymes with making phosphorothioates and having to ligate unnatural nucleotides as well. So this seems simplest. It also gives us what we believe to be the best separation of intermediates in terms of molecular weight from the product. So we'd make that product separation slightly more straightforward. When we demonstrated in the lab, we wanted to really use the template interaction to purify our final product. Obviously, shorteners of the correct sequence, the only ones that will really bind effectively to the template. And so we used shorteners synthesized by liquid phase, and they were not purified in any way at all. The ligase enzyme's undergone several rounds of directed evolution to get up to a suitable activity to use in demonstration. And during our process development, we managed to take a reaction of this, at the end point here, where we've got some remaining shorteners. This is where our intermediates appear. We've got a product, and this is our hub template right at the end. And we've managed to improve that reaction through to something which looks like this, where we've pretty much consumed all of our shorteners. We've got very little intermediate left, and this is about 95% conversion of our starting materials all the way through to the product. Taking this into the process, and this is 5-litre chemistry we did in the labs in Stevenage. Again, this is our lovely big vessel with our enzymatic ligation going on. Once it's heated, then all protein falls out of solution and can be removed by depth filtration. Ultimately, we end up with a very clear solution here, this is necessary actually for the ultrafiltration. We don't want any fines in there which will precipitate out and blind our filters. So we then go in, run our concentration. pH 11 is what we use to purify away our shorteners and intermediates, and then we recover the API at pH 12. Once that's done, take back to pH 7.6 to recycle the hub template, and we're able to desalt, lyophilize our lovely clear solution of oligo to give this beautiful white solid at the end here. Each of those trays carries about 10, 12 grams of oligonucleotide product. What we have found during purification is that sounds very straightforward. It's not quite that straightforward. We always get a little bit of permeation of hub template with our API fraction. But this can be removed by a second pass, giving us actually an incredibly clean product here. So to think that we're getting, by HPLC here, up to 99% purity, starting from some of our inputs around 85%, shows the degree of purification that we can get from using this template interaction. In slightly greater depth, looking at that purity, then what we find is these synthesis failures n-1, n+1. These are things which carry through from our starting materials. And we very closely track where they come to being right at the ends of our strands, and we're not getting any sequence failures, as it were, in the middle of the oligonucleotide. What we find is that we are very, very closely controlling what happens in the middle of the oligo. In terms of the control, this end actually is very often controlled for us at the moment by the chemistry, but we do have some variation at this end. The other impurities we have to concern ourselves with are not immediately evident by the HPLC alone. We have to think about things like phosphate impurities, which will appear underneath our main peak, and they're detected by LCMS, so not shown here. But these, again, are present in our starting materials. They do carry through our ligations, and they end up in the final product. But these are all impurities which are known, and they are actually part of standard chemical synthetic methods, and so they are all qualified for use through the material that we are making by chemistry. So we are, with the exception of this intermediate here, which we control by our process, there is nothing else in here which is not produced by chemical methods of oligosynthesis. Now, I mentioned that we do have, at the 5' end, a certain degree of flexibility of what we can put there, and we can use that to our advantage, because not all of the oligos that we make are just simple oligonucleotide strands. Some do carry functionalization at the end. So in the case of another example we've been working on, we have this tris-galactosamine unit, which is appended to the 5' end. Can we put this into a ligation reaction? Well, yes, we can. We did actually purify this shortmer in this example, and we did this really to remove any of the ungalnacked material from it. So we have a nice clean source there. But all the other shortmers, again, were unpurified, as we had done in our antisense campaign. And what we find is the enzymes will tolerate this very well. In fact, in the lab, we're seeing, for the sense strand, which carries the GalNAc, very similar process yields to what we get with the antisense strand, which does not carry the GalNAc. So we really think that we are in a good position here to use this to synthesize modified oligos as well as just plain oligos as well. So that really brings me to the end of the chemistry here. This is something that we've been developing for, well, I've been involved with it for about 5 years, actually. The things that we've been most impressed with have been actually how easy it has been to develop and run. And the biocatalysis works really, really nicely in the stir tank vessels. We've had a lot of learning to do with the purification, because the whole ultrafiltration processes have been very, very new to all of us. It's not something we do very much when we're working with traditional small molecules, so there's been a lot to pick up there. But at the end of the process, we are getting this absolutely exquisite purity in our APIs, which we think is something that you can't get from traditional chemical methods, and they all use chromatography as well. So this is all in development. At the moment, we do think that as we get improved methods of synthesis of shortmers in particular, we will be able to use this to deliver much cheaper oligos, much higher quality, much more sustainably. So just to acknowledge some of the key contributors to this project, this has been a really, really multidisciplinary project across the board. I sit in process chemistry. It's my job to take these and scale up these reactions, but there's a lot of work that goes on in enzyme development and evolution, in all the analytical work and that kind of thing, and actually learning to work with people who have different scientific skills and learn from them is something that I think goes all the way back to the work that I did during my PhD, where we started as chemists. I came in as a trained chemist. And Jim was very keen that we picked up new skills. It sent us down Tennis Court Road to the biochemistry laboratories to learn how to purify proteins and so on and so forth. And it was this morning when I was putting this together, I came across this photo in Alison's, from Alison in the memory book. And here we have actually a prime example of Jim encouraging somebody to do something they have never done before. So here he is. Exactly how we got here, I'm not entirely sure on this trip. There's quite a possible chance that there was pretty poor pre-task analysis went on, which led to somebody falling in a ditch they didn't know existed. But yeah, here we have Jim encouraging his PhD student to do something he didn't know this was going to work. He thought this was fine. But he's really there, front and central, encouraging somebody to develop a new skill that they never knew they had. And it all turned out well. He's still in the audience, so it's all okay. But I think it's something that we all owe to Jim, that he encouraged us to be open to new things and to learn new things. And for that, it's something I've had to carry through. In industry, we have to chop and change what we work on all the time. We have to be agile and be able to move. And it's one of these skills I think I'm indebted to learning during my PhD. Thank you very much.